Okay, we have quite a few people who have joined, so we're going to go ahead and get started. Good morning, everyone. Thank you for joining our Swede Midge webinar this morning. We're excited to share some new research results as well as some other management and biology information today. Just wanted to get started with a few housekeeping announcements. We do encourage you to stay muted during the presentations just to limit some feedback and background noise. We will be recording the presentation, so for those who are not able to make it today, we will email out the link to the recording at the end of today or early next week, and then we'll also post it to our Eastern New York YouTube page. We do encourage you to keep your video off just because we are recording and it can cause a little bit of distraction. There are some points during the webinar when we do encourage you to interact with us. So there will be some Q&A portions where we do invite you to unmute if you'd like to ask us a question. You can input your questions through the Zoom chat box, or you can raise your hand and unmute yourself, and we can have a discussion there. There will be plenty of time for Q&A, and we really welcome you to interact with us and hope to be able to answer all of your questions. We are offering New York State DEC credits for this webinar, pesticide recertification credits, and that protocol was in the event registration page as well as the email. And just as a reminder there, you will have needed to email me a photo of your license. I will need that by the end of the day. Quite a few people emailed those to me already or texted it to me. And if I received it, I will have responded. If I didn't respond, please send that to me again, just in case I didn't get it. We are requiring you to complete a pre-program Qualtrics survey to sign in for the DEC credits. You can access that survey through the chat box. Jen is going to put the link in a few times for people. And looks like she just did that now. You can also scan the QR code there that will take you to the survey. It's a very brief sign-in survey. You'll just need to input your name and your certification number so that we can verify your identity. We'll ask you to do that at the end of the program, and you will need to stay online for the whole program, and then we'll email a certificate out to you. Just a brief overview of our agenda. We have a few pre-programmed survey questions to ask you before we get started to just get a sense for how much people already know about Swede Midge before our program. We'll review the biology and management of Swede Midge, and then we'll delve into some research updates on different management strategies. We'll have an overview of research activities from the University of Vermont, manipulating the chemical ecology of Swede Midge. We'll take a short break around 10:15 so you can get up and stretch your legs, get some coffee, etc. And then we'll hear from Christy on her ground barrier work to suppress Swede Midge emergence. I will end the webinar with some case studies and what we've learned working on individual farms to manage Swede Midge. And then we'll end with some extra time for Q&A and then another post-program survey. I'd like to take some time now just to introduce the speakers before we get started. I can go ahead and get started. My name is Elisabeth Hodgdon. I'm a regional vegetable specialist with the Cornell Cooperative Extension Eastern New York Commercial Horticulture Program team. I'm based in Plattsburgh, New York, and I've been working with Swede Midge since 2015 when I was a student in Dr. Yolanda Chen's lab at the University of Vermont. And I'll kick it over to Yolanda if you'd like to introduce yourself next.
Everybody, my name is Yolanda Chen. I'm a professor in the Department of Agriculture, Landscape, and Environment at the University of Vermont. And happy to be part of this event. And it's really nice to have a check-in. I've been working on Swede Midge for about 16 years now. So unfortunately, progress is slow, but still steady.
Thank you, Yolanda. Also working in Dr. Yolanda Chen's lab is Vincent Tamakloe. Would you like to introduce yourself?
Hi everyone, I'm Vincent Tamakloe. I'm a PhD student, and like Elisabeth said, I'm in Yolanda Chen's lab at the University of Vermont. Yeah, that's it.
Thank you, Vince. And Christy.
Christy Hoepting. I am a regional vegetable specialist with the Cornell Cooperative Extension Vegetable Program in Western New York. And I've been working on Swede Midge since 2002, starting with early detection work. And I'll tell you more about what I've been up to in my presentation later.
Okay, thank you, Christy. And I know in our audience group, we have some researchers and some growers who have been working with Swede Midge in one way or another, either through research or managing it on their farms for a number of years. So we welcome you to share your expertise in the chat or to unmute yourself during the program. We'd love to hear about Swede Midge from you as well. Okay. Oops, sorry. So we have a few pre-programmed quiz questions, and no stress here. We're just interested in understanding more about what you already know about Swede Midge. But before we get started actually in these, I wanted to ask a question about where about everyone else's role in the audience. So I'm going to go ahead and launch a quiz question now, so bear with me. So what is your role? Are you a farmer? Are you an extension educator or specialist? A researcher? Do you belong to a government agency? Are you an other ag service provider? So for example, from a soil and water conservation district or other, and I believe you can select all that apply. And just as another little icebreaker, I'm curious to know where you're from today. So if you could put into the chat where you're located. I know we have folks from Canada, folks from around the Northeast and the Midwest. Where are you joining from today? Okay, yeah, quite a few folks from New York. We have Quebec, Canada. We have Michigan, New Hampshire, Maine. Yeah, so thank you all for joining today. It's a good mix of folks from different roles as well. Okay, so our first quiz question is, which of these photos depicts Swede Midge? And I'm going to go ahead and launch the next poll for that. We encourage you to um, answer through the Zoom poll feature. But if the poll, for whatever reason, isn't working for you, you could always answer in the chat. And I'm going to wait to go over the answers at the end of the program. Leave it up for another couple seconds here. Okay. So most folks selected photo B. My next question for you, which photo depicts Swede Midge damage to a crop? Having a little difficulty launching the poll. Let me see if I can go back here. All right. I'll ask just for a little help from Jen. Jen, are you able to close out the poll that's being shared? Ah, there we go. All right. Which photo depicts Swede Midge damage? So A is showing some curled leaves on what looks to be a Brussels sprout plant. B is showing a scar in place of where a sprout would be on Brussels sprouts. C is showing hollowed cauliflower. And then D is showing some swelling of the roots in what looks like a cauliflower plant. Okay, move on to the next one. So this is a statement about Swede Midge biology that we're going to ask. Does Swede Midge overwinter as an adult in trees along the field edges, as eggs in crop debris, as pupae in the soil, or none of the above? Okay. We just have a couple more questions here. Which of the following is an effective management strategy for Swede Midge? Insect exclusion netting, barn-wide crop rotation, ground barriers, or all of the above? Okay, and then the last question. We are curious if you would be willing to try pheromone mating disruption to manage Swede Midge. And that's a practice that we'll be talking about later on in the program today. Okay, well, that's about it for the polls for now. So thank you for participating in that. And next, I am going to share my slides for the next presentation. Okay, everything look okay on your end?
Looks good.
All right, thank you. Okay. So now I'm going to give an introductory presentation on Swede Midge, focusing on what crop management practices are effective for this pest, crop damage symptoms, a little bit about biology, before we delve into the new research. So just a bit of a story about Swede Midge invasion in North America. Back in the 1990s in Ontario, Canada, there were vegetable producers who were seeing some really odd symptoms in their brassica vegetables. They were seeing broccoli crowns that had twisting and scarring within the florets, as you can see on the left. They were seeing plants that had swollen petioles and twisting near the meristem. And then there were some plants that had a dead meristem in the center, what we would call a blind plant on the right. It turns out that was that was due to an insect called Swede Midge, which is a small galling fly. You can see in the photo that it looks a bit like a mosquito with long legs. It's very small, 2 millimeters in length, and because of its small size, that caused some difficulties in its initial detection and identification in its invasive range. It has what we call a cryptic or hidden feeding behavior. The larvae are feeding within the meristem of the plant. And so because of this hidden behavior and the small size, it's difficult for farmers sometimes to know that they have Swede Midge until the population increases to into a large population. This is an invasive species that is native to somewhere in Europe and Asia. The exact origin of the species is unknown, but it's present throughout most of Europe and most parts of Asia. It was identified in Ontario, Canada in 2000 and has been confirmed in many new states and provinces since. And it infests nearly all of the brassica crops that are important to us. So broccoli, cauliflower, red Russian kale, those tend to suffer the most economic damage. It's also a big problem in canola production in Canada. This is its current invasive range, unless there's been a really new documentation of the species that I'm not aware of. In the United States, the latest additions to the map were eastwards in Maine and then west to Minnesota. It hasn't spread as far south as maybe what I would have expected. It's been mostly spreading east and west. In Canada, we don't think it's found further west than Ontario. There were some initial detections in the Canadian prairies. That's why those provinces are shaded in kind of a speckled red. But come to find out, the insect causing damage to canola in the prairies was actually a newly discovered species, canola flower midge. Like I said, we don't think that Swede Midge is further west than Ontario and Canada. Back in 2018, when I was working with Dr. Yolanda Chen's lab, we did a survey of producers in the United States and in Canada to ask them how much they were losing due to Swede Midge damage. And it was kind of staggering the amounts that some people were reporting. So on average, over $3,000 per acre per year because of Swede Midge in their brassica vegetables. And at the time, a lot of the serious damage was reported kind of in river valleys in Vermont, Quebec, and in some areas of New York. And since then, I know that there have been folks reporting serious losses in Maine, in Minnesota, in the Midwest. But some of the more severe losses historically have been closer to where Swede Midge was initially detected in North America. I'm going to go over a little bit more about the biology of the pest and something to know about Swede Midge. Again, why it makes it such a difficult pest is that the adults are laying eggs in the growing point of the plant. Those eggs hatch into larvae that are feeding kind of hidden within the growing point. So in this picture on the right, this person is peeling back the small leaves in a cauliflower meristem, and you can see the yellow larvae feeding there. They have kind of a unique feeding habit in that they feed through extraoral, they secrete saliva that digests the plant and then suck it back up. And that manipulates the plant growth and causes deformities. So is a lot less conspicuous than, say, a caterpillar on a brassica plant because it's difficult to see them and the damage doesn't show up right away. The damage typically can be visible in a plant about 2 or 3 weeks after larvae are feeding. And by the time the plant is showing those damage symptoms, the larvae will have left the plant to pupate in the soil. And Dr. Chase Stratton led a study a few years ago looking at the damage threshold for Swede Midge in cauliflower. And he found that it really only takes 1 to 3 larvae per plant to cause a cauliflower to not produce a marketable head. So this is a really low damage threshold. Again, something that makes Swede Midge difficult to manage. I'm going to show a few more pictures of what Swede Midge damage looks like. So this is damage in Brussels sprouts. You can see on the left there's some curled leaves, some scrunched leaves from larval feeding. What tends to cause the most damage is when Swede Midge kills the apical growing point of Brussels sprouts so that it can't continue to grow upwards to produce more sprouts. It can also cause damage to the individual sprouts on the Brussels sprout stalk. So on the right there, there's a good clear picture of a brown scar where there would have been a sprout, and the Brussels sprout is not there. This is what damage looks like in kohlrabi on the left and cabbage on the right. You can see the kohlrabi bulb on the left is scarred, and that, of course, would not be marketable. This can sometimes be confused with slug damage, but if you don't see a slug and are seeing these scars, that's likely Swede Midge damage. On the right is a cabbage head that had been damaged by Swede Midge when it was newly forming. So this is what we call kind of like a fluffy cabbage head where it doesn't produce a nice, dense, normal head that's marketable. This is what damage looks like in kale. On the left, the growing point has been damaged and the plant may or not be able to grow out of it. Kale is usually able to grow out of a lot of Swede Midge damage, but the leaves may be smaller, there might be multiple growing points, and you have these twisted petioles that may or may not be marketable depending on who you're selling to. So the next growth stage of the Swede Midge is the pupae, which reside in the soil. So the larvae jump off the plant when they're ready to pupate. They bury themselves in the soil and they pupate there and then emerge about a week to 14 days later, depending on the weather. In the fall, increasingly larger number of these pupae will form cocoons and then stay there over the winter. So Swede Midge will be emerging the next spring where they were pupating, which is where you had an infested crop last year. And that's an important piece of Swede Midge biology to know when you're planning your crop rotations. Another little tricky thing about Swede Midge is that the pupae can remain in the soil for up to 3 years. Most of them are emerging the following spring, but there is a small percentage of them that can stay in the soil for 2 or even 3 years, which is an evolutionary strategy for them to kind of hedge their bets with, say, if there were adverse weather conditions in that next growing season, some of them are waiting to emerge until the next or the year after. It really helps to use monitoring on a farm to know if you have Swede Midge. It's really helpful for initial detection if you're wondering, do I have Swede Midge? I've been seeing some weird damage symptoms in my brassica crops. It also helps to visualize what we kind of refer to as an invisible pest. Otherwise, the adults are really small, they're difficult to see, and having them stuck to a sticky card on a trap can help better understand the population on a farm. So individual farms can set up traps in their fields, and it helps to work with an extension agent if you need some help with identifying Swede Midge. They can be a little tricky to identify since they're so small. So a Swede Midge trap, the trap system that works best for this is using what we call a Jackson trap. It's a delta trap, small cardboard, has a sticky insert, and you'll put a pheromone lure inside. The pheromone lure is a plastic cap that has been formulated with a synthetic version of the female sex pheromone. And that pheromone will only attract male insects. And so sometimes with monitoring, people get concerned that the traps will actually bring in the pests to infest their crops, but really it only attracts the males, which don't lay eggs on the crop anyway. I mentioned that identifying Swede Midge can be a little tricky. What you're really looking for are a certain number of segments on the antennae length and shape. You can see it looks like it's beaded and has some hairs on the individual segments. You're also looking at the veins, and there can be some lookalikes that can get stuck on the traps. So it does help to work with somebody from your government or extension if you have some questions about identifying them. And then if you are living in a state or a province where Swede Midge hasn't been documented before, it is important to get those insects identified through molecular means so that you can be absolutely sure that you have Swede Midge and we can record it on our distribution map. So I'm going to review some management strategies for Swede Midge, and unfortunately there aren't a lot of options out there, hence wanting to do more research on different strategies. So for conventional farms, synthetic insecticides are an effective tool for managing this pest. There are a number of products that are labeled for Swede Midge nationally. Unfortunately, OMRI-listed insecticides are usually not effective. The timing of them can be difficult, and because they don't have systemic activity, they don't necessarily reach the insect that might be kind of buried in the growing point. If you think back to the photo I showed of the larvae, it can be difficult to get good contact with the larvae if they're underneath some of those leaves. Insect exclusion netting can be an effective strategy, and Christy has done quite a bit of work with that. I'll talk about that in a bit. There hasn't been much luck with investigating host plant resistance. There have been some trials looking at broccoli variety, tolerance of Swede Midge, cauliflower, collard greens, and there are no good recommendations for example, grow this broccoli variety because it is tolerant to Swede Midge. That unfortunately, we don't have any great recommendations for that. There are some crop types that are less susceptible, and we'll talk about that in a bit. Trying to bury the pupae really deep in the soil has had some mixed results. Generally, it isn't an effective strategy. We do recommend disking in and burying crop residue. That is an effective strategy to prevent Swede Midge from building on an old crop. Crop rotation can be very effective as long as you're manipulating space and or time. There's been some research on biological control from Canada, and they have not yet identified an insect that is able to reduce the population enough for it to be eligible for commercial distribution. There is a wasp that they found that is naturally occurring in Canada that has some effect on reducing Swede Midge and canola systems, but we don't know yet if it's present in vegetable systems and doesn't decrease the population enough for it to be available commercially. And then Christy will talk more about ground barriers in a bit. I'm going to talk about prevention for a bit now. Sorry, just catching up on the chat. I'm sorry if some people are having problems hearing. I'm going to talk a bit about prevention. So it's really critical to start the season with a good crop rotation in mind. I'll mention some specific recommendations for crop rotation from Christy in a bit. So essentially moving your current brassica field away from where you had infested brassica crops the previous year can be effective for managing Swede Midge. It can also be effective to skip spring brassicas. So if those insects are emerging from an infested site, and if the adults don't have any crops around to lay their eggs on, that can crash the population and taking advantage of isolated fields. It's important to use clean seedlings. I worked with a farm that a few years ago that hadn't had Swede Midge on his farm before, And then he had purchased some seedlings from an area in Vermont that had Swede Midge, and we believe that those seedlings were infested when he brought them over. And this can happen when farms are purchasing seedlings from each other, when they're not protected in the greenhouse, or if they're hardened off outside when there's Swede Midge flying around. This can start you off on the wrong foot in the season if you are planting those seedlings that have Swede Midge larvae on them. We recommend destroying infested crops immediately after harvest. So, for example, a broccoli crop, when the main crowns are harvested, if those plants are sending up multiple side shoots afterwards, some folks will harvest those for their markets. But if you're just leaving them in the field for an extended period of time, Swede Midge can use those side shoots as hosts, and that can grow the population. So we do recommend disking in, tilling up that crop after you're done with it. And also, this is a little easier said than done, but cleaning equipment in between fields, especially if you're going from, say, an infested field to a new field that has never had brassica crops, you could transport those pupae in the soil. And that's something to think about if you're purchasing tillage equipment from another farm. It's a best management practice to make sure that that's really clean so that you're not bringing in insect pupae and weed seeds. I'm highlighting this graphic from Christy's crop rotation fact sheet, and maybe Jen can pop that link into the chat. We'll also email you out some links to the resources that we're mentioning in our follow-up email. So Christy is highlighting some creative ways to use crop rotation to manage Swede Midge here. And the initial recommendations for crop rotation for Swede Midge, I believe, were about 1 kilometer distance in between fields, which of course is not realistic, especially for a lot of small scale growers. So what Christy has found is that you can take advantage of some features in the landscape to reduce that distance. So that could be rotating in field or planting brassicas in fields that are separated by trees because the insects have difficult time flying through woodlands to get to their next host. It could also mean skipping spring brassicas so that the midges are emerging in the spring. There's no host plant for them to lay their eggs on, and then the population crashes. So I encourage you to take a look at that fact sheet, and I think it helps a lot with thinking creatively about spatial and temporal crop rotation. I should also mention that Swede Midge can use weeds as hosts. So there are a lot of brassica weeds that are present on farms, mustards, shepherd's purse, etc. Those can sustain a small population, but not to the extent that economic crops can. This is another graphic from Christy's fact sheet looking at different preferences for crops. Starting at the highest risk for loss, that would be broccoli. Red Russian kale is the most preferred type of kale. Cauliflower, kind of in the middle would be Brussels sprouts and cabbage. Cabbage is susceptible when it's young, but then once it has a few leaves covering the growing point, that is inaccessible for Swede Midge, so it's less or not susceptible after a certain stage. And then least preferred would be some quick growing crops like radishes or baby leaf brassica greens, and then Savoy cabbage and Chinese cabbage. So thinking about crop rotation, you should also take into consideration crop preference because it might be possible to grow a least preferred crop type in an area where you know you have Swede Midge emerging. Again, these are some photos from Christy. Christy's done quite a bit of work on insect exclusion netting. This can be very effective for Swede Midge as long as you are installing the netting over uninfested ground. So that would be ground that hasn't had brassicas for at least a couple of years. Otherwise, if you're setting the netting on top of soil that has pupae in it, those would be emerging and then they're contained with the crop, which is what you don't want. And so you need netting that has a pretty fine mesh size, so 25 gram. Protect Net is a material that we've recommended to a lot of growers, but there are other brands out there as well. The system can be kind of expensive initially, but most of the materials can be reused, and the netting can be reused for several years as long as you're taking care not to rip it, or if you're, you can also mend those holes in the netting too. I'm not going to spend too much time talking about ground barriers because Christy will go into that at length, but we have some new data showing that it is possible to smother an emerging Swede Midge population. So laying a landscape fabric or silage tarp over it in the spring will prevent those adults from emerging. And we would recommend applying that over ground from early May through mid-July. And we have some data suggesting that this can be highly effective. A number of years ago, well, not a number, a couple of years ago, the Swede Midge researchers that are on the call today, we put together a comprehensive review paper of Swede Midge management strategies for organic farms. And that is open access, so freely available to anybody with the link. So maybe Jen can put the link into the chat too. So it's in there now. So if you'd like to read up on some of the management strategies I mentioned and some others, that's a good resource. And we put together this decision tree for organic management of Swede Midge, which I have here. And that highlights some of the most successful management strategies that we know have worked for farms. So if a farm has enough space for spatial crop rotation, we would recommend that. If it's a small scale farm, there are a few other options such as using insect exclusion netting, using temporal crop rotation or skipping certain brassica crops in the year to crash the population, and perhaps growing some less susceptible brassica crops. And we're hoping to maybe add ground barriers to this in the future, and perhaps also nematodes, which is something else that we've been working on. I wanted to talk about materials for conventional farms. And these are products that are labeled in New York State, and I believe they have national labels as well. So historically, neonicotinoids have been relied upon heavily by farms. I know that there's been some talk of phasing these out. Some of these have become restricted use, whereas they were not before. So there are two nicotinoid products. Warrior is another option. And then there are a couple others, Movento and Senstar. And what makes some of these products effective is that they have systemic activity, which is important to reach those larvae that are kind of deep in the growing point. And some of these materials are really useful in spray programs for brassica growers because they have some they're effective against some other pests as well. So for example, Movento, systemic activity, some of these are effective on cabbage aphids, caterpillars, etc. So historically, Swede Midge has become, it is difficult for conventional farms to manage if they've let the populations increase by quite a bit, either through early on in Swede Midge invasion in North America. It was difficult to identify Swede Midge, and a lot of folks didn't know what they had. So if populations were really large, synthetic insecticide programs could still fail. At low populations, these can be highly effective in managing the crop, especially in conjunction with things like crop rotation, and perhaps growing some less susceptible crops in infested areas. And I'll end the webinar later on today with some specific examples from case studies. So we've been working with individual farms over the years, and just wanted to highlight some of those practices that I mentioned before. So really, farms that are successful in managing Swede Midge are identifying the damage and confirming the pest early on so that the population doesn't become large. They may be using some sort of what we call a brassica break to disrupt populations. They may be using systemic insecticides to suppress populations, using crop rotation. And then increasingly, we're working with more farms that have been able to successfully adopt ground barriers and netting and some other experimental strategies. And I wanted to end there to see if anybody has any questions. I know we have a couple of minutes before the next presentation starts. So I'll just pause here to see if anyone has any questions, and I'll stop sharing. And our next presenter will be Yolanda. So I'll invite Yolanda to share her screen while we're gathering some questions.
Hi, Elisabeth. I have a question. Um, Thanks so much for the presentation. And during the prevention slide, you mentioned in the greenhouse, like seedlings can be infected. Does that mean like if they used infested soil, or how does that seedling get infected?
Yeah, that's a good question. Thank you. It Usually it's not infested soil because folks are typically using new potting mix for their seedlings, although it could be possible if they're um, using other soil. It typically happens if you have Swede Midge adults that are flying around that come into the greenhouse. So for example, if you have brassica transplants after mid-May when the spring adults have emerged, those can actually get into the greenhouse and lay their eggs on the seedlings. A lot of farms will set out their transplants, say on wagons or benches outside to harden off before transplanting. So if there are midges in the landscape, they can lay their eggs on them then as well. We've set up some Jackson traps inside propagation greenhouses and have actually been able to trap adults in there, which kind of shows that they're able to come in if the greenhouse isn't completely enclosed.
Great, thank you.
There's a question in the chat about pesticide treated seeds being effective. I don't know that that would be applicable to vegetables, but I'm not sure about canola. Maybe Christy has more information on that.
I do not know of the use of seed treatments in vegetable brassicas. They would be for cabbage maggot primarily, but at a 5-week-old transplant with a seed treatment, the seed treatments are running out of gas. So that's not how we treat for cabbage maggot with seed treatment in brassicas, or at least in transplanted brassicas, I'll say that. Maybe in direct seeded. So if you had an insecticide for cabbage maggot that also had activity on Swede Midge in a direct seeded crop, that could be effective, but probably not in transplants. I don't know anything about canola.
Thank you, Christy. The next question is, how are they controlled in Europe and Asia? So I believe they're kind of using the same strategies that we're using over here. Because this was kind of a new invasive species in North America, there were instances where the populations built to really large levels on farms in Canada and the United States, and it kind of seemed like the problem was more severe in North America, but it certainly is a problem in Europe and Asia. And I haven't really communicated a lot with researchers over there. So again, I invite Christy or Yolanda if anybody else has any insight on how they're managing it over there. But there are still researchers over in Europe who are looking at control strategies for this pest. So it's definitely a problem there as well.
There are a lot more natural enemies attacking Swede midge in Eurasia, and so that's why it's not as severe of a pest. I believe when they looked at parasitoids, these are wasps that lay eggs and feed internally within the Swede midge as sort of like parasites. I think there are about 7 species. None of them are very, dominant overall, but I think this is why it hasn't been a focus in Europe.
Yeah, thank you, Yolanda. These are some good questions. If you have any more, if you think of them during the presentations, you can feel free to put them in the chat then. But otherwise, I'm going to turn it over to Yolanda and Vince for the next presentation.
Okay, great. Well, thank you, Elisabeth, for organizing this. Is really a treat to be able to share with you what we've been working on, like I said, over the last 16 years. You can see there's a lot of storytelling around kind of a lot of missteps or kind of dead ends along the way. But this has been really A lot of people have been involved, and that's why just on my first slide, I want to just show some of the students that have been working with me over the years. So Elisabeth, working on pheromone mating disruption. Now Vince is also working on pheromone mating disruption. A former student, Chase Stratton, who's now at Delaware State University, working on repellency. So let me just, okay, there we go. So what's challenging about Swede Midge is that the larvae feed in a very unique way. Like Elisabeth mentioned, they secrete saliva. And within the saliva, it's a little bit of a kind of a mystery as to what exactly is the key ingredient that basically causes a plant to freak out. So the saliva is kind of like basically they spit on the or vomit on the plant, and then they suck up the juices. Within the saliva, there seems to be genes that are called plant effectors. They have the magical ability to stimulate the plant to grow in specific ways. There's the possibility that the midge is also producing plant hormones itself. It's also kind of has digestive enzymes and other things like microRNAs. So kind of information or instructions that may lead to kind of this distorted plant growth. So the problem really is that the plants are, the midge knows kind of the key to manipulating the plants, and the plants react badly. And that's what leads to the unmarketable produce. So as Elisabeth mentioned, we have been trying to understand, is there even a threshold for some of the more susceptible plant varieties like cauliflower and broccoli? And we found that kind of there's no threshold that works for kind of the brassica oleracea crops, for the heading ones, just focused on broccoli and cauliflower because they're the most susceptible. And as a result, you know, what maybe many of you are familiar with, like, you know, the typical IPM strategies of scout until there's a certain threshold and then spray. This doesn't work because, first of all, as Elisabeth mentioned, very small larvae, you have to destroy the plant to even find a single larva. And then also kind of by the time you see the damage, it's too late. So as a result, we have tried all sorts of different strategies to try to figure out how do you crack the code, basically? How do we, with an interest in managing Swede Midge, what are all the possible approaches that we can take? And we've had, I guess, generations of undergraduate students that have come through and graduate students, including, say, Katie Jacob, working on things over 10 years ago. And recently, Elisabeth led a paper, which there's a QR code here also, which summarized all the work that we've done, including everything that was unpublishable because there were no positive results. And this is where we kind of took everything that you can find, say, within the textbook, you know, typical pest management textbook, and we said, well, how about this? How about this? How about this? And that's why this particular article has been kind of a major milestone, is that it allowed us to kind of like assess where are we at. So, so you can see here from this figure, there's been a lot of strategies to try to target Swede Midge, trying to understand where it's most vulnerable in its life cycle. The figure shows kind of all the different strategies that we've tried. The asterisk shows kind of what seems to be partially effective or is promising or is effective. And you can see the list is pretty limited given the amount of effort that we've thrown at this. Okay, so because we found that there's essentially no threshold, we've focused on preventing damage and focused on the key thing is to prevent oviposition. And you could think about that life cycle as what are all the things that we can do to prevent either mating or prevent the females from laying on eggs on the plant. So are there aspects of repellency that we can explore further? So Chase Stratton asked the question, are more genetically different plants more repellent? Can we take odors from other plants that are really completely unrelated? And so this on the left side is the angiosperm phylogeny, which is basically the large family tree of how all plants are related together. He found that plant essential oils, so the things that you see at the health food stores that are basically the key chemical compounds responsible for the aromatic properties and the odors. He found that the ones from more like different plant families tend to be more repellent, and he found that garlic and lemongrass were the most promising. Some of them, if you sprayed them directly on the plant, ended up causing phytotoxicity. He followed up with some trials and found that somehow in the field, garlic essential oil was not as effective, but he used the product Garlic Barrier and found that it reduced damage by about 20%. But this is something that we, want to follow up more before we can really make this a true recommendation, because 20% is not that great. And you can see here, like there's still some scarring on the broccoli head. This is one of our trials, but overall, at least there's still a head versus kind of a blind head. So we've also focused a lot on this aspect of trying to prevent mating, because this has been an effective strategy in other systems, and we've been trying to figure out how we can adapt that to Swede Midge based on its biology and ecology. So Elisabeth worked on her on this topic for her thesis, and at that time the biggest complication was that the idea of pheromone mating disruption had only really been successfully used with moths, caterpillars. So how pheromone mating disruption works is that you can, once you identify the chemical compound that the female produces, you can produce that and release it at very high levels, 1,000 times more than the typical female might produce. And so males are then kind of, they perceive females everywhere because the scent is everywhere, but they're unable to follow the trail because there is no trail, because the air is so saturated with the female sex pheromone. So they think they're in heaven, but then the idea is perhaps they get exhausted trying to chase after these, the odors, and they cannot find the trail. And the idea is that they die just sort of like, after exhausting themselves. So at the time when we were doing research, fly pheromones were very expensive. And this is because if you look at this figure here, there's this 2S, 9S, 2S, 10S. This is because the attachment of the some of these other subunits of the compound were in a particular three-dimensional orientation. And if you had the mirror image of it, they would not work. So moth compounds, the pheromones, are much simpler. They are just, say, a straight chain without all these extra complicated, appendages there. And so as a result, this made fly pheromones very, very expensive. And so just as an illustration of how expensive, we received a grant for $250,000. My first quote to get, a significant amount of this pheromone was going to cost $250,000. We thankfully found Chemtica, which is which actually makes the active ingredient for the Salida pheromone traps that Elisabeth was just talking about, and we were able to kind of progress with the study. So what we found, what Elisabeth found, is that basically males are attracted to both natural and racemic pheromone blends, and so we could have used this. The racemic blend is the mirror image compound. So by doing that, she found that the cheaper version of it, which is that there's two orientations, both of them would work for the purposes of attracting males. So, so she also found that these compounds, when they were saturating the airspace, they could also prevent males from finding females and mating with them. And so these are things that she did in the lab. She also found that in the field, sometimes it also reduced damage in the field. The challenge, though, with interpreting too much from this field study is that she did her work in Ontario, where the midge was everywhere. These are very large fields of brassica fields. And she was only kind of isolating kind of a small 16 by 16 meter plots. So there's plenty of opportunity for midges to mate outside of the areas that we were directly treating. So the fact that we even found that there was the potential for marketable yields gave us hope that there is something to work towards, and we could progress that further. Oh, this is incorrect in that this is already been published, and somebody could put that link in the chat as well. So following that, one of the biggest challenges around pheromone mating disruption is that it's only been successfully implemented usually in high value tree fruit crops, so apples, pears, walnuts, or in vineyards. So this is where we know year after year exactly where those insects are going to overwinter, and we know where the plants are going to be. So cycle after cycle, they are dropping to the ground, they go and pupate underground, and then they emerge again. And these are long-lived perennial plants. So the plants, we know exactly where the plants are. The problem is with a system like kind of brassicas, cold crops, is that we keep moving them around. And so because of that, one of our grants that we finished up and we're in the midst of publishing the publications from it, we've been asking the question, well, where does actually Swede midge emerge and mate in the landscape? We want to know how long it stays in the soil. And so if you can imagine, if you're rotating across multiple years, the farm could be this checkerboard of Swede midge emergence sites. So this is work that was led by Andrea Campbell, or Andrea Swan at the time, and she is currently writing up her research, and she was asking these questions of where does Swede midge mate, what seemed to be kind of the primary drivers of kind of this interest in mating, and how long do they remain in the soil? So we want to figure out, do males, when they emerge from the soil, do they just hang around? And Elisabeth had noticed that they did. And then, or do they just take off and they fly and they're kind of scanning around to find like an odor or a pheromone trail emitted by the female? So what Andrea's found is that the Swede midge appear to mate at the soil surface in the last year's field, and that they've remained in the soil. Most of them emerged the following year. So what we did is we took a bunch of pupa that we reared in the lab, and then we buried them in the field, and then we looked at kind of over 3 years what percentage emerged. And it looks like most of them emerge in that first year. And so because of this, what this is setting up us to do is to kind of design an entire system around the Swede midge biology. And you can see it's complicated enough. It's not, a lot of this kind of outreach and discussion is really important to kind of figure out, well, how can we implement this? Is this going to be commercially viable? So there is potential for Swede midge pheromone mating disruption. We've found that now there are cheaper precursors that have lowered the cost to produce the pheromone. And I believe the pheromone kind of price per acre now is about $75. The challenge, though, then is how do we deliver it cheaply? So when we've done it, they've been in small little vials and baggies, and we're putting them actually every 2 meters apart, which is definitely not commercially viable at all. We understand that. We also been looking at this idea of where can they actually be placed, the pheromone dispensers be placed? Should we put it in last year's field or this year's field? And those are some of the questions that my current student, Vince, will be kind of following on further. So some of the things that have kind of prompted our, this wave of research is thinking about how do we actually lower the cost of dispensers? There's new technologies for kind of releasing things. And we are working with Chemtica, the chemical synthesis company, to kind of try to figure out, like, how do we take, either materials, where do we try to release it, what are the different kinds of dispensers, with the goal of trying to come up with something that is affordable, that just, it's kind of like you apply it and leave it, and also hopefully trust it. Okay, so Vince, you can go ahead and take it, and just let me know when to forward the slides.
Yeah, so for me, over the summer of 2025, which is just last year, I was trying to look at evaluate the two dispensers, the bag and then the paste. So this is basically the same formulation, but different mode of delivery. So I was trying to evaluate the efficacy and the durability of these two dispensers in the field to manage the Swede Midge. So the motivation is that while the bag dispensers is effective, they can be labor intensive, as Yolanda has mentioned. And then also this can limit the practicality of growers. Therefore, we wanted to evaluate whether the paste formulation can provide a simpler and equally effective alternative to the bag. Let's go. Next slide, yeah. Yeah, so to do this, we were asking the question, can the paste formulation disrupt the male Swede Midge as effective as the standard bag dispenser? And so we went out into the field, and then we had like a design where in that design we were trying to look at the bag versus the paste, and then we also had a control plot. So in a broccoli field, we have 3 plots, and then in that 3 plots, one of them we were putting the bag dispensers, the other we were putting the paste dispenser, and then the 3rd plot, which is the control, we didn't have any pheromone in that field or in that plot at all. And then in each plot, which is a 6 meter by 6 meter plot, we were putting the dispensers, 8 dispensers in a staggered grid format, which you can see with a plain background with some dots in it. And then in the middle of that plot, we put out the pheromone trap. So the pheromone trap has, which you can see below here, the broccoli field with a white pipe and then a trap hanging on there. That's where we put the pheromone trap. And then the dispensers are already placed in the field. And so whilst they emit the pheromone in the field, the pheromone trap will kind of has the standard pheromone or dispenser that Solida produces, and then it attracts, and then we see how many males in the field will be attracted to that particular trap. In that particular trap also, we have a sticky card that when the males are attracted by this pheromone, they get stuck on the trap, and then we pick the sticky card, bring it into the lab, and then count the number of males that are on that trap. We did this for over 5 weeks, and then the metric for this is looking at the trap shutdown. And when we say trap shuts down, we are looking at what happens when the what happens when the ambient pheromone levels in the field are so high that males cannot distinguish between pheromones in the trap, and then the resulting is very few males are captured in the trap that is placed in the middle of the field. Let's go. Next one. Yeah, so after deploying this dispensers in the field, and data was collected over 5 weeks, we were looking at each week when we bring the dispensers into the sticky liners into the lab to kind of count the number of male Swede Midge that were collected by this trap. We were looking at in the first week, for instance, you could see there is so much uh, males that were collected by the control trap. And then in the control trap, this is the plot which did not have any pheromone at all in there. And then the blue and the green is referring to the bag and then the paste also. You could tell in the first week, the pheromones that are released by these dispensers were so high. And so very little of male Swede Midge were captured on those plots. And then as time progresses, the numbers kept reducing and reducing. At the end of the day, we realized that the trap shutdown for the bag or the sachet dispenser was 86.2%, and that of the paste is 88.2%. So that means the number of males that were captured in the bag or dispenser plot has reduced by 86.2%. And then on the paste plot, it has reduced by 88.2%. And then looking at trap shutdown, the threshold is if the reduction is 80% or above, then it means that the dispensers that we have put in the field is actually effective and then it's working. So I also look at the comparison between the bag and then the control and then the paste and then the control. And then you can tell that there is so much difference between the bag and then the control. But between the paste and then the bag, there is not much difference. As you can see, 86.2% and 88.2% is not much difference. And so this is just telling us that the paste formulation, which is not supposed to be as labor intensive as the bag, is also yielding the same results that we expected. And then also I was looking at the weathering or the bioactivity of these two dispensers in the field. And then when I talk about the weathering or bioactivity of these dispensers, I'm looking at when we put the bag and then the paste in the field. And then after the rains, the sunshine, the wind blowing and all of that, how long are these dispensers going to stay in the field at the same time being effective? So what we did was that we put 60 of each of the dispensers, that's the bag and then the paste, in the field. And then we left them in the field for like 12 weeks. Every 2 weeks, we bring in 10 dispensers, 10 of the paste and then 10 of the bags into the lab. And then we kind of expose them to the male Swede Midge in a wind tunnel. So the wind tunnel is the photo you see at the bottom right corner. We put the dispenser in the tunnel, and then there is a fan on one side of the tunnel. And then when we put the dispenser there, at the other side, we release male Swede Midges. And then the fan is put on to blow wind through the tunnel. So as the wind is being blown through the tunnel, the plume that is coming from these dispensers are being released into the tunnel. And then we see or we count how many of those male midges fly towards the dispenser, the position of the dispenser. When the males are able to fly to the dispenser, then it means that there is still some plume there that is attracting the males onto the dispensers. And that is the bioactivity we are talking about in this situation. Yeah, and then looking at that, how those dispensers in the field are attracting the males when they are brought into the lab. You can see in the first week before we took the dispensers onto the field, we kind of did an experiment to see how effective or how many Swede midges, male Swede midges they are attracting. So like I said, 10 Swede midges are exposed to each each dispenser. And then in the first week, before the dispensers are even taken to the field, we realize that it is attracting about 90% of the male. So out of 10 midges that are released in the tunnel, about 9 is being attracted to the dispensers. This shows like a high that the dispensers are highly effective. And as you see the trend on this graph, after every 2 weeks, when we bring the dispensers in the field and we do the same protocol, exposing the midges to the dispensers, we realize that the numbers of the midges that are fly towards the dispensers kept reducing. So, but even in this situation, the idea is to see whether the paste dispenser will still work as effective or equally to the bag. And then we realize that yes, it worked equivalently to the bag, but slightly a bit maybe a little more effective than we thought it would, because in at the 8th week we realized that the bag dispenser had a sharper decline, and then the paste dispenser had like more steady kind of decline in its attractiveness to the sweet midges in the field. So from this graph, we can conclude that the paste dispenser offers field durability that is comparable to the sachet technology that is being used there. Yeah, so the take home from these two experiments that were done is, one, the paste dispensers perform equivalently to the bag or the sachet dispensers in the field trap shutdown efficacy trial. And then also the paste dispenser showed comparable durability to the sachets under the weathering experiment also. Now, both traps also meet the threshold for trap shutdown. That means in 4 weeks that they were deployed in the field, they were having a trap shutdown of over 80%, which is the threshold that shows how effective the dispensers are. But one thing that I also notice is, because we are trying the paste bags or the paste dispensers for the very first time, I also wanted to see if the paste dispensers can go beyond the 4 weeks that the bag dispensers are known to be effective. And so I went on to do like a 5th week, taking data from the field to check if it will exceed the 4 weeks duration. And then I realized that after the 4 weeks, in the 5th week, the trap shutdown declined sharply. For the paste dispensers, it was recording around 59%, which shows that in the 5th week or beyond the 4 week period in the field, the paste dispensers will not be as effective as we want it to be. And even much worse was the bag dispensers. It recorded about 29%. So the threshold, which is 80%, that shows effectiveness. If you are having a trap shutdown of 59 and 29%, that is not an effective control strategy that we want to put out there. So the recommendation will still be that dispensers in the field should be replaced after every 4 weeks to maintain a strong mating disruption in the field. Yeah. Yeah, these are the
Okay.
2 experiments I did over the summer.
All right, thank you, Vince. Just where we're at right now is we still have research trials planned. We are going to be testing how kind of actually whether pheromone mating disruption can actually lead to higher marketability of overall kind of broccoli plots. And this is something that we'll be doing this summer. Actually, we have a very complicated experiment in multiple sites. Where we're at right now is we're still focused on figuring out how to lower the pheromone delivery costs. The paste is just one type of dispenser. There are other ones that might even kind of be pheromone impregnated, I don't know, sort of clay-based balls or, wax-based balls. A lot of these materials have the potential to be certified by OMRI, but they're definitely kind of products that are going to be very different than I think, what most people are used to, kind of thing. And so we're very aware around kind of the labor costs, and the ease of use is really critical for figuring out what would actually be, more widely adopted. So feedback from all of you would be really helpful on this. And we think that there's other lines that are potentially promising. So say like garlic barrier, we've done some trials. There's some partial kind of benefits from that, but definitely for a lot of this, it's taking the effort to continue raising the funds to do research, more research. With that, just want to thank my lab. We have different sources for funding. And if you have any questions, and if I have any time for questions, I'd be happy to answer them. I can look at the chat. I saw a bunch of different questions. The one question about sterile males is definitely a possibility, but typically that takes federal interest and support. So say like the screwworm for fly, that's been kind of a bilateral or multilateral cooperative effort to kind of release that. And the difference is, say, for the screwworm fly, if anybody's heard of that, that particular insect mates once a year, versus Swede Midge, it's constant. It's every the life cycle, probably the heat of the summer is every 2 weeks kind of thing. And so you constantly need to have males around. So that's why it's not the most ideal for this, because it's over these multiple overlapping generations, and adults only live for 2 days. So it becomes a little unwieldy, I think, for that approach. Let's see. I saw Elisabeth asked her question on mass trapping. Were these studies done in large plantings of a single crop? So yeah, so for a lot of our trials around like the pheromone delivery work has been in fields. In Vermont, we do not have very large fields. These fields are about kind of an acre or two in size. But the advantage of being in a place where we have many different small fields is that we are going to be able to kind of create local infestations. We're not saying exactly where they are. And then also testing whether pheromone mating disruption actually is occurring. So we're doing things in sort of isolated regions that allow us to say, okay, simulate kind of pheromone mating disruption in isolated areas and whether it's actually effective in protecting vegetables. Pheromone mating disruption has been attempted in some other systems. And one of the key things is trying to figure out that isolation and then also kind of where things are. So in the systems where it has been successful, the growers are planting the same crop in the same field year after year, which is not necessarily, best practices, say for, soil fertility and rotation and disease management practices, but there's other reasons why growers are doing that. I see one other question. Bark beetles have a pheromone that prevent aggregation. Swede midge have a similar phenomenon. Not that we know of similar pheromone. It does not have an aggregation pheromone. Bark beetles aggregate because they kind of collectively, they're so small and they're trying to take down a large tree. So that's why they're kind of releasing these aggregation pheromones to seek out buddies to collectively overwhelm the tree. Swede midge does not do that, but one of the things that Elisabeth found is that females actually can auto-detect. They can detect the female pheromones as well, and it actually stimulates them to double their effort to call for males. So that's one of the things we know, that chemicals are really important for communication. And so they are able to kind of sense that as well. I think I'm out of time, definitely.
Yeah, thank you, Yolanda.
Okay.
We are cutting into our break time, but I wanted to keep the conversation going because I thought these are some really good questions and answers. I just wanted to add another quick thing about the auto detection is we've looked at whether the females are attracted to their own pheromones, and we found that they're not. So the female pheromone that we've been using for monitoring doesn't actually attract the female, so it doesn't work to aggregate both sexes of the population. So with that, we have about 8 minutes for a break. So feel free to get up, leave your computer, stretch, refresh your coffee, use the bathroom, etc. And we will rejoin at 10:30. In the meantime, if you have any other questions, feel free to drop them in the chat. And Christy will be your next presenter.
Thank you. Sorry I went over.
Okay, thanks, Yolanda and Vince. Okay, it is now 10:30, so we will resume our Swede Midge webinar, and our next speaker will be Christy Hoepting to talk about her work with ground barriers. So take it away, Christy.
All right, thank you so much for giving me this opportunity to share my research with you. So I am a senior extension educator with the Cornell Vegetable Program in Western New York. And I first met Swede Midge in the United States in 2004 in Niagara County as part of our early detection survey. As Elisabeth mentioned, it was first detected in Ontario, Canada in the year 2000. It took 4 years to spread to the United States. And then it took about 10 years after that to disperse and spread within New York State to the point where we started getting complaints from organic growers about Swede Midge. So from 2015 to 2017, I had a project that included intensive monitoring of Swede Midge on small scale organic farms using the pheromone traps to really show the growers what this quote unquote invisible pest looks like on their farm and how the population pressure relates to the damage they're seeing in their crops and their management practices. And I will say that managing Swede Midge is as unique as each farm is unique. It looks different on every farm, and it's really helpful that the growers can see their pest population build and crash. And so we've actually started up a very similar project right now with Elisabeth and Lori Connick, and we're trying to get with the Northeast Air Grant, we're trying to get 10 farms through this monitoring program and set them on their way so they have figured out what makes sense for managing Swede Midge on their farm. And also part of that project, 2015 to 2017, I did a lot of small plot on-farm trials looking at trap cropping, garlic essential oils as a repellent, insect exclusion netting, as well as several other on-farm experiments. And it is from that work that one of my growers asked me, what about using landscape fabric or silage tarp or even just the black plastic mulch that we're using as a ground barrier to prevent Swede Midge from emerging as adults as a way to crash the population. And that concept was intriguing to me. And so I literally pivoted my research in Swede Midge to focus on trying to understand whether ground barriers had potential as a management strategy for Swede Midge. So one of the questions that we had was, is the Swede Midge, are the Swede Midge adults going to emerge underneath the ground barrier, or are they just going to lay in waiting until you remove the ground barrier? And so, as I mentioned, we were interested in plastic mulches, landscape fabric, and silage tarps. And all of those create different conditions underneath the ground, our ground cover that could affect emergence of Swede Midge. So I started in 2018 and 2019 with what I call small cage studies, and my treatments included an untreated check, so no ground barrier at all, just bare ground, biodegradable black plastic mulch intact as well as with holes, landscape fabric intact as well as with holes. And the question there was, can you dual purpose plastic mulch or landscape fabric for preventing Swede Midge from emerging, but also growing a crop at the same time? So, for example, onions in landscape fabric. And then we looked at silage tarp intact because that's used for weed management. We're not planting a crop through that. So 6 treatments, and then you can see them here, the bare ground, a tarp, landscape fabric without holes, with holes, and then the plastic mulch. And the small cages were handmade, 2 feet by 2 feet by 1.5 feet high, covered with Swede Midge insect exclusion netting, trap inside, infested ground. And we had 6 replications, so we had total of 36 cages out there. And the way we infested the ground was working with Yolanda and getting infested cauliflower plants from her Swede Midge colony. And that first year, the timing of when we got those infested plants, they had already started dropping to the soil to pupate. So we just took the top 1 inch of the pot with the cauliflower plant in it, incorporated that 1 inch into the soil, grabbed that growing point in case there was any larvae in there, getting ready to drop to the soil, and then put the ground barriers on, put the cage with the trap in over top. So here's the results from that first year of study. And so we're looking at the different treatments across the x-axis, and then we have 2 sets of bars. We're looking at the total number of Swede Midge captured per cage, the average of those 6 reps. So the first bar is what we captured from July 20th to September 4th, that 48-day period where the ground barriers were on. And the second bar is what was the number of Swede Midge that were captured after the ground barriers were moved from September 4th to October 25th, a 51-day period. So in the bare ground, we captured 55 Swede Midge in that 48-day period. And then we caught less than 1 Swede Midge in all of the intact ground barriers. However, you can see clearly in blue here that we caught an average of 33 Swede Midge, significantly less than the bare ground, but still probably way too many to be effective as a management strategy when we punched holes into the plastic or the landscape fabric. So that would suggest that unfortunately you can't dual purpose the ground barrier and put a crop over top of it. The Swede Midge adults are going to emerge under there and they're going to find the holes and they're going to escape. And then when we look at what came out of the ground after the barriers were removed, for the most part, nothing, including in the bare ground. Truthfully, most of those Swede Midge emerged within the first 2 weeks. However, we did catch 1 Swede Midge in the the plastic mulch with holes, and 4 Swede Midge after the tarp was removed. And so even though these results appear to be very encouraging that ground barriers could be very effective at crashing a Swede Midge population, because this trial was running so late into the season, I question whether my trap catches were low because the ground barriers were effective, or is it because they were going into diapause and getting ready for winter and they weren't going to emerge anyway. So in 2019, we repeated the trial, this time a little bit earlier in the season, so we didn't run into issues with diapause in the fall. And so this time, the infested plants that I got from Yolanda, the Swede Midge, were a little bit younger. So we had to plant the whole plant on June 29th inside the cage, wait 11 days until they had dropped to the soil to pupate. Then we just severed that plant at the soil line, grabbed the growing point, watered it in, then put the ground barriers on and set up the traps. And so here's the results from that study, identical to how I presented the 2018 results. So we can see the ground barrier from the period of July 10th to August 9th, that 30-day period, we captured 21 Swede Midge in the average of the 6 replications in the bare ground. We caught less than 1 Swede Midge in all of the intact ground barriers. And again, we see the Swede Midge emerging through the plastic mulch and landscape fabric with holes. So pretty clear that you're going to have to take ground completely out of production, can't dual purpose by putting a crop over top of the ground barrier. And then when the tarps were removed from that period of August 10th to September 25th, that 47-day period caught absolutely no Swede Midge in the majority of the treatments, except less than 1 in the plastic with holes and the landscape fabric with holes. But overall results extremely encouraging that intact ground barriers have potential to manage Swede Midge. So the next step was to figure out how is this going to work in the real world. And this is some of our trap catch data from spring emergence sites from that monitoring project 2015 to 2017. And you can see we're getting a bimodal emergence with a peak occurring end of May, beginning of June, and then a second peak occurring in June. And then the emergence starts to really drop off. Um, our first trap catches are second half of May, all zeros on the 13th of May. These traps are set up the first week of May, end of April. And then we're still getting some lingering Swede midge popping out of the ground, but these are open air traps. So this is a little bit muddy because we could be catching first generation adults as well. But nonetheless, what we are looking at is to shut this down with the ground barriers to prevent these high numbers from spewing onto your farm and causing trouble in your brassicas. And I just wanted to point out, this is trap catch data from Emergence traps, so repurposing those small cages. This was a site that was heavily infested with Swede midge in the fall. They planted a cover crop, and then this is first thing in the spring, and we're measuring the Swede midge that come up out of the site just in that 2x2 area. And of course, our trap catches are much lower, but we're seeing that peak flight, and then still Swede midge are popping out of the ground through July into August, but the numbers are very, very low here. Probably not going to cause economic damage. We're really looking to shut down that peak emergence. So the next trial that I did, I call it a large scale study. And the treatments that I had included an untreated, so just bare ground, no ground barrier. And broccoli was planted on the 22nd of May, harvested on the 21st of July. So the broccoli was exposed to Swede midge for 63 days. And then tarp and landscape fabric moved forward. We left black plastic behind because that's just too fragile, too likely to tear, not as durable as what we need for a ground barrier. And of course, we're not putting holes in them. And so we're basically evaluating the tarp and the landscape fabric with 3 removal times, including the 27th of June, the 21st of July, and the 15th of August. And we're removing the ground barriers 18, 25, 28 days after the broccoli was planted, and then harvesting another 30 days later. So the broccoli has approximately 30 days of exposure to Swede midge after the ground barriers were removed. And the ground barriers were established on April 21st in this trial. But before that, we had to establish Swede midge infested ground with overlapping generations to stimulate what growers would be experiencing in the real world. So we moved to a new research site. The large cages were 4 feet by 4 feet by 4 feet using these electrical conduit hoops covered in insect exclusion netting. We planted in that 4 by 4 foot area 9 plugs of red Russian kale. And then we introduced 2 Swede midge infested colony plant, or 2 Swede midge infested cauliflower plants from Yolanda's Swede midge colony. That went out the 3rd week of May. And then we just let that trial go and fester with Swede midge for the entire growing season. You can see the kale is getting quite big in the middle of the season there. And then in the beginning of 2023, when we set up the ground barrier study, this is why you don't leave exclusion netting out over winter, because it gets trashed in the snow and ice. But you can see the remnants of the kale, and these are the infested. This is our nicely infested ground. And then what we did is dig a trench for the ground barriers to ensure that they were secure, that they're not going to pop up. And you can see a bare ground, landscape fabric, and tarp. And then we replicated those 7 treatments 4 times. And then the next thing we did was plant broccoli right beside that infested site. So we extended the cages 8 feet, put in 10 broccoli plants, put an emergence trap in so we can track what's coming out of the ground, an open air trap inside that cage, and then just let the broccoli grow, become infested with Swede midge. And when it was time to remove the ground barrier, we just simply cut that out, very easy. And then when the broccoli was ready to harvest, we evaluated Swede midge damage. And so here are the results from that study. Here we're looking at the average weekly trap catches across the 4 replications for the different treatments. And so this one here is bare ground. So you can see that bimodal peak coming out of the ground, and then it starts to drop off a bit. And then so this is for the duration that the broccoli was exposed to Swede midge. Bare ground is always color-coded in brown. Tarp is always color-coded in black. Landscape fabric is always color-coded in yellow. And so here we see the trap catches after the landscape fabric and the tarp is removed at that early timing, June 27th, which is color coded in green. And then the same thing with the July 21st removal, color coded in blue, and the August 15th ground berry removal in color coded in purple. And so you can easily see that for the duration that the broccoli was exposed to Swede midge after the ground barriers were removed, we caught a little bit of Swede midge with landscape fabric with the June 27th removal, practically nothing after that, but we are seeing Swede midge pop out of the ground after the tarps were removed. So here's a summary of that data, and to ensure that it's all the same units, we are looking at the total number of Swede midge captured per day. And here's the bare ground. And I flanked off what's not significantly different than the bare ground. And so what you see is that the landscape fabric treatments significantly reduced Swede midge trap catches by 81 to 98%. We're not really seeing much difference between the July 21st and the August 15th removal. And although the June 27th removal is not statistically different, numerically, we captured 4 times more Swede midge. And then you can see, obviously, we have quite a few trap catches following the tarp removal. Generally not significantly different from each other. For the most part, not significantly different than the bare ground. And so my theory here is that the Swede midge are laying and waiting underneath that tarp, and then when it's removed, that's when they pop out of the ground. And here I just wanted to show you the emergence trap cages. So these are the upside down laundry baskets that we use. So a very small area, but the traps are not being contaminated with first generation Swede midge coming off of the broccoli. So this is just what's coming out of the ground. I'm just going to show you the trap catches from the bare ground treatments. And we see that peak emergence and then it dwindles off, but there's still a few Swede midge popping out of the ground. Here we have a resurgence at the end of August, which was only in 1 rep. This is an anomaly. I think that cage must have been compromised. So if I take that trap away, this is what the bare ground would look like. And then here I added in the trap catches from the tarp. So you can see in green, the June 27th removal, the July 21st removal in blue, and the August 15th removal in purple. So definitely Swede midge coming out of the ground. Our traps are not contaminated from anything coming off of the broccoli. And then here is the landscape fabric emergence trap catches. So we're getting something following the June 27th removal, but practically nothing following the July 21st and August 15th removal. So now let's take a look at the damage on the broccoli when we harvested it. So I use a 6-point scale for damage in broccoli heads. So a 0, no damage at all, perfectly healthy heads. 1, minor damage. This is where if we dig for it, we can find some damage, but it doesn't affect yield or quality or marketability. 2, minor moderate. Now we can find damage if we dig for it. It could reduce quality or yield, but a consumer would not readily notice that damage and still purchase that head of broccoli. Moderate, now we're moving into unmarketable territory where that head of broccoli just does not look right. You can see scarring, it's disjointed, it doesn't look healthy. Moderate, severe, now we're into a severely distorted or scarred head, and severe is a plant with a blind head, no head to market at all. So 0 to 2 are in the marketable category. So here we're looking at Swede midge damage. On the left, this is the percent of plants that have damage, scale 1 to 5. And this on the left is the mean Swede midge damage severity rating per plant. And so here's the bare ground, 77% damaged heads, and here's the bare ground averaging a 1.7 severity rating. So that's coming in at minor moderate. And what we notice right off the bat is in green here is that when we remove landscape fabric and tarp on June 27th, we had 92 and 100% unmarketable plants and a significantly higher severity of head damage than the bare ground in the unmarketable category. So June 27th removal was too soon. And then we look at The later removals in landscape fabric, no significant difference between the July 21st and the August 15th, practically no damaged heads at all, and the damage rating is very, very low. You could totally market all of that broccoli. Tarp, we're seeing more infested plants. In side-by-side comparisons, we have higher damage with tarp than we do with landscape fabric, and we have significantly less severe damage in the heads compared to the bare ground with the tarp. So, and then here we are looking at just the percent of marketable broccoli heads. So the bare ground has 70% marketable heads, and what we see with the June 27th tarp and landscape fabric removal, less than 50% marketable heads. Absolutely unacceptable. But everything else is doing really, really well. 100% marketable heads with both of the landscape fabric treatments. No difference between the July 21st and the August 15th removal. And the same thing with the tarp. 95% and 86% marketable heads. No significant difference between the July 21st and August 15th removal. So, to summarize these results, with respect to the Swede Midge trap catches following ground barrier removal, the landscape fabric was more effective than the tarp. So, compared to the bare ground, the landscape fabric significantly reduced Swede Midge trap catches by 81% when the ground barrier was removed on June 27th, by 96% when the ground barrier was removed on July 21st, and by 98% when it was removed on August 15th. No significant differences among them with respect to trap catches. Now, with the tarps compared to the bare ground, no significant differences, but numerically saw a reduction 13 to 42%, no significant difference among them. And I suspect what's happening is that light and moisture is getting through the landscape fabric, which is triggering the Swede midge to emerge, but then they perish underneath the landscape fabric because they can't get out. And then alternatively, the tarp doesn't let moisture or sunlight through, so there's nothing to trigger the Swede midge emergence. When you remove that tarp, they see the light, they get the moisture, and they pop out of the ground. With respect to the timing of removal of ground barriers, June 27th removal was too soon for both landscape fabric and tarps, even though the trap catches of Swede midge was so much lower with the landscape fabric, it was still too high to result in economically marketable plants. And then we didn't really see any difference between July 21st and August 15th. Ground barrier removal was effective for both landscape fabric and tarp. Landscape fabric a little bit more effective than tarp. But at the end of the day, we had 100% marketable heads with the landscape fabric and 86 to 95% marketable heads with the tarp. So what remains unknown is like what would happen if the ground barrier was removed on July 7th or 14th. So June 27th was too early, July 27th, and of course anything after that was fine. What's the breakdown in between there? Well, unfortunately, I have not had the opportunity to trial that. And then the other question is, does the removal date depend on the year, which really does it depend on the weather conditions because the weather is different every year. So that remains unknown. But based on the research results that I have, my recommendation would be to use landscape fabric to prevent Swede midge emergence. It needs to be applied before Swede midge start coming out of the ground, so late April, early May. And I have to say that it's got to stay on there until the third week of July because I know the end of June is too soon, and I don't know where that cutoff is in the middle, so I'm going to say the third week of July. And of course, anything later than that should be fine as well. You need to ensure that the ground barrier is secured so that Swede midge cannot escape. So we can't dual purpose it and plant a crop in it, so we're not punching holes into it. But also if you're using sandbags to secure it, you got to make sure you have enough of them that a wind isn't going to rip that tarp up and let the Swede midge out. And I would prioritize the placement of ground barriers where you have a heavily infested site or a brassica planting that was heavily infested the previous fall, because that's going to be your spring emergence site. And then you're going to have to be mindful of other nearby sources of Swede midge in determining whether ground barriers are worthwhile and whether susceptible brassica crops may be grown on the site. So this is going to be very farm specific. So if you have a 2-acre field, for example, half of it was in brassicas, season-long brassicas, let's say, you probably don't have to worry about the spring plantings that are harvested in July. There's probably not going to be a lot of Swede Midge coming out of the ground there. You're going to probably have some from the summer plantings. And say you had 300 rows of broccoli that was heavily fested in the fall. You could only market less than half of those heads. You could cover that up with tarp, and then depending on what the pressure is in the rest of the brassicas, you may not want to plant brassicas on that site until the spring emergence has subsided. So using crop rotation in addition to crashing that heavy population with the tarps. Or you might decide, I'm gonna take ground that hasn't had brassicas in it for over 2 years, and I'm gonna plant something in the spring and I'm gonna put exclusion netting over it. Or I'm gonna plant less susceptible brassicas on that site. So you have to like take everything that's going on into making these management decisions. And I assume if you're using mating disruption, you're asking the exact same questions. Where am I going to put the mating disruption? Where's going to give me the most bang for its buck? Because you're not going to treat half of your acreage with a ground barrier or with mating disruption. And so it's going to be part of an integrated approach to Swede management. So the next steps were to take it on farm, do on-farm demonstrations and see how this is working in the real world. And so this is where I passed the baton to Elisabeth, who worked with farms in 2024 and 2025, implementing ground barriers, and she'll share those results with you momentarily. So before I just wanted to mention funding for this project. I had to piece together little bits of funding from different sources just to get this work done. Fortunately, Cornell's Toward Sustainability Foundation gave me some money 2018, 2019 for the small cage studies. I got some money again in 2022 to establish the overlapping generations of Swede Midge in preparation for the large scale study. And then Yolanda and I worked together to get a Northeast IPM partnership grant. That helped fund the small scale studies. And then in 2023, to do that last step of evaluating the ground barriers, I got a CVP challenge grant for the supplies. But definitely lots of in-kind contributions from my team with respect to labor and mileage. And speaking of in-kind contributions, have to highlight Yolanda, um, for being my Swede Midge supplier. It costs money to rear her colony. And I'm very thankful for her for giving me Swede Midge so I could do this work. Also, the Cornell Lake Erie Research and Extension Lab in Portland for free hosted the 2018 small scale study. Adam McAllister is a garlic grower at Holly who hosted the second year of the small scale study or small cage study. And then Andy Harrington's Produce in Oakfield has hosted all of the large scale studies. And I'm still working there on that farm. We've transitioned from groundberry work to studying entomopathic nematodes. And then finally, the several technicians over the years that have worked on Swede Midge with me. And if there's any questions, I just wanted to point out search Better Know a Pest Swede Midge is a 1-minute video on how to find the Swede Midge larvae in your brassicas. And then again, our crop rotation recommendations for Swede Midge also includes the relative preference of the different crops. That's available online as well, and that came out of the monitoring project from 2015 to 2017. I believe there's time for questions. And Elisabeth, do you mind if I stop sharing my screen?
Yep, that's fine. Thanks, Christy.
Okay.
There is one question in the chat that you could answer. The question is, could spot treatment of gaps with pesticides in landscape fabric be a viable option for a home grower?
So using the landscape fabric to prevent Swede Midge from emerging, but spraying an insecticide over the holes, is that? Or spraying it between beds? Let me look at the chat. If I can find that question.
Looks like they elaborated over the holes.
You would have to, I mean, you're going to be targeting flies that are emerging at different, I don't know that you could target the adults emerging from the holes very well. Do you have any other thoughts?
That would be my answer as well. I think
Yeah,
the timing of it and the practicality would be difficult.
right. Yeah.
We do have an extra minute. I'm going to go ahead and get my PowerPoint lined up, but if anybody has any other questions, feel free to unmute or put them in the chat. Thanks, Christy.
Thank you.
Okay, hopefully that looks all right on your end. I'm going to give the final presentation for our webinar just with some take-home messages about lessons learned working with individual growers to manage Swede Midge. And as Christy mentioned, managing Swede Midge, there are a set of effective practices that we recommend, but every farm is different. So each farm has a unique landscape and crop rotation pattern, might be organic or conventional. Each farm is unique. And we work with growers to consider all of those different factors and come up with an IPM plan for them. I mentioned the decision tree earlier on in my presentation, and that really highlights the most effective strategies that we've helped growers adopt over the years. And one takeaway for me is that it's seldom the case that only one strategy can be used to successfully dampen a Swede midge population. This pest is difficult for several reasons that we highlighted earlier in the program. And I think this is probably the case for other pests as well, that thinking about the pest life cycle and adopting multiple practices to disrupt the Swede midge life cycle is what's really allowed some farms to successfully manage this pest. And so that could be crop rotation, temporal and spatial, foregoing specific plantings of brassica crops when populations have built up, using netting if you're farming in a smaller space, foregoing highly susceptible crops in favor of less susceptible ones, and then using synthetic insecticides, and also using those preventative measures that I talked about before. So using uninfested transplants, post-harvest crop destruction, and then using monitoring as a tool has been helpful as well to visualize this pest. This isn't a terribly exciting diagram, but I wanted to use it to illustrate a point that Swede midge has multiple life stages, and the different practices work to suppress their transitions through these different life stages. And I think that it's helpful to recall information about the pest biology and behavior when you're trying to think about what might be effective on your farm to suppress those populations. So we've talked about using ground barriers. Christy just talked about using ground barriers. So that disrupts the pupae from the adults from escaping from the soil after they finish pupation. So that's kind of targeting one portion of the life cycle. We've talked about multiple practices that can prevent adults from laying eggs, and Yolanda highlighted that in her presentation, and Vince talked about mating disruption as well, that we really want to prevent the eggs and larvae from being on the plants in the first place, because once they're there, it doesn't take very many of them to disrupt the plant's growth and to cause heading brassicas to be unmarketable. So we discourage that by using crop rotation, post-crop destruction, netting, mating disruption potentially. And then we have conventional insecticides that are helpful tools to kill the larvae that are in the plant. We've highlighted the importance of monitoring, and I encourage folks who are on the call who maybe aren't sure if they have Swede midge, but maybe have seen some suspicious symptoms, to work with your local extension or governmental service with trapping. We're happy to help with giving recommendations on trapping. It can be a little daunting to adopt at first because identification of the midges can be difficult and requires a hand lens and knowing what you're looking for. But certainly I hope you feel empowered to start a monitoring program on your farm so that you can better understand the population dynamics and then use that information to create your IPM program. Wanted to highlight some individual farms that we've worked with over the years and their successes. So one farm that I worked with that participated in our ground barrier study, they lost all of their fall broccoli in 2023. They were kind of at their wits' end with dealing with Swede Midge. It had been on their farm for a number of years. They worked with us to adopt ground barriers in 2024 and then use them again in 2025. The photo on the right there, these were the beds where they had their fall brassicas, and they reported nearly 100% yield loss there. They had to abandon most of the crops. So we covered that with landscape fabric from late April through late July. We used monitoring around the farm to better understand the population. And the farm also used row covers in the spring, mostly for flea beetle management, but that also helped keep Swede midge off of the brassicas earlier in the season. They've pivoted to growing some less susceptible brassicas, so Napa cabbage, some quick growing brassica greens, and bok choy. Those have been popular in their for their customers. So they've pivoted from growing crown broccoli to growing some of those other less susceptible brassicas. And I'm kind of excited to share that in 2024 and then 2025, they were able to successfully grow fall sprouting broccoli with little to no damage. And so it's been some work and attention related to Swede midge, but using a combination of these strategies has really helped them crash the population. Another farm we worked with was in kind of a similar situation where they reported near total loss of their brassicas in 2023. What worked for them, because brassicas were not as brassicas are not essential for their markets. They were able to take a complete break from growing brassicas in 2024. And we do recommend this if you have a high population and need to crash it. So they pivoted to other crops. They resumed brassica production in 2025, and we measured relatively low levels of Swede midge in monitoring. And last year, they're able to grow a nearly perfect fall crop of heading broccoli, crown broccoli, which I thought was a great success. And I thought that they did a good job in choosing different management strategies that would allow them to nearly completely crash the population. But one thing that surprised me a little bit here, well, maybe it's not surprising, but I think realistically, a farm probably can never completely get rid of a Swede midge population. So even though they completely got rid of their brassica crops in 2024, there were still some on the farm in 2025. So again, a portion of those midges emerges a second year, which was happening there. Something else that was kind of unique to this operation that was surprising is this farm acquired a second vegetable production field down the road, separated by quite a bit of space and trees and houses. And we were trapping for Swede midge there. That field had zero history of vegetable production in the past. We couldn't find any brassica weeds there, and we still caught Swede midge there, which was a little discouraging. But what I think might be happening there is that they could have been moving Swede midge pupae from their main vegetable production space into that new space on tillage equipment. So this pest is really difficult. There are multiple ways that it can survive despite management strategies, but using a combination of these practices, this farm was successful in growing brassicas last year. I wanted to highlight some takeaways from our SARE Research and Education Grant, and Christy and Lori have been working with urban farms for a few years now. And so I thank Lori for highlighting some of these successes and the farmers who participated. So what we found to be successful on urban farms in particular, so there have been a number of cases on urban farms in different states of Swede midge where, because of the lack of space for rotation, Swede midge can become a big problem. So insect exclusion netting has been a good practice for those farms in managing Swede midge. And I've kind of been harping on the fact that you can't use netting over infested ground, but But what some of these farms are doing are putting netting over infested ground, but then growing a non-host crop underneath. So that's allowing them to use the infested space and prevent the midges from moving, which I thought was a great kind of creative strategy. And taking advantage of different features in the landscape for crop rotation, either foregoing certain plantings, taking advantage of secluded or fields that are separated by buildings, and then monitoring, working with Extension, holding some educational activities for community gardeners to increase awareness for this pest. We've highlighted these resources throughout the program, but I wanted to highlight a couple of different ones that I don't think we've mentioned so far. One is that under Yolanda's USDA Crop Protection and Pest Management Grant, I've been working on revising the Swede Midge Information Center for the US website, which was initially created by Dr. Tony Shelton and Julie Kickert and Christy were some of the original authors on the website. And I've been working on updating that with new information on management strategies based on our research, as well as an updated list of references that folks can turn to for more information. There are a few other updates on distribution. And so Jen just shared the site in the chat. Tune into that later on. It should be finished pretty soon, hopefully. And we're going to include an interactive feature on the website with research updates, which I think will be kind of fun for folks to follow along as we hopefully learn some new information that can be useful for others on Swede Midge. There is an organic management of Swede Midge fact sheet that we wrote a few years ago that we are going to be having translated into different languages. So if Extension service providers are working with audiences who would benefit from having that fact sheet in, or I believe Spanish, Nepali, French, and remind me of the other language, but those should be released hopefully sometime later this year. Arabic was the other one. Thanks, Lori. And we will put our contact information in the chat at the end of the program, so feel free to reach out to us if you have other questions, and we'd be happy to help you with IPM decision-making and identification, et cetera, if you believe you have Swede Midge on your farm. And we encourage you to reach out to your local extension service for assistance with monitoring, because again, that can be a useful tool. So with that, I'm going to stop sharing my screen for this presentation and again pause to see if we have any questions.
I just wanted to make a comment that as you've relaunched the Swede Midge Information Center site, one of the things that people can do as part of audience participation is if you search for Swede Midge through a Google search, and then you go down and you click it, that will help boost the page rank to kind of get it sort of resurrected as kind of a really current and up-to-date place for information.
Yeah, thank you, Yolanda. I also wanted to pause, and people can share in the chat or you can unmute yourself. I know there are a number of farmers on the call who have experience with Swede Midge, and also I invite others to participate. Is there anything that we missed in our presentations that you feel like have been helpful for you to manage Swede Midge? Is there any expertise that you would like to share? I invite the audience to share your words of wisdom on Swede Midge as well. I'm curious what has worked for you. Okay. If anybody has any thoughts later on, again, feel free to put those in the chat. But if there's no other questions or comments for now, we are going to move on to the last couple of parts of our webinar. So thanks for tuning in up until this point. We are going to revisit our quiz questions on the same questions that we asked earlier today, and we're hoping to measure what you learned about Swede Midge today. We're going to ask this question again, and bear with me as I pull up the poll. Which photo depicts Swede Midge? It looks like most people have answered. I'll just give folks another few seconds to choose. And again, no pressure. You're not going to be held, it won't be held against you if you get the answer incorrect. Okay, so it looks like most folks got the answer correct. This is showing Swede Midge here with B. The photo on the left, A, is actually cabbage aphid. You can see it's more kind of dusty looking. If you look closer with a hand lens, you would see legs. The photo C is showing flea beetles. That's a pretty obvious difference in IDing those versus Swede Midge. And then D is a diamondback moth caterpillar. Much more conspicuous than Swede Midge larvae. Going to move to the next quiz question, which is identifying Swede Midge damage. Which photo depicts Swede Midge damage to a crop? So again, A is showing curled leaves and Brussels sprouts. B is showing a scar where the sprout would be. C is showing hollow growth in a cauliflower head, and D is showing some swollen, deformed roots in a cauliflower. I'm going to go ahead and share now. So the correct answer to that one is B. I showed a photo of this earlier today. In case you're curious, photo A is leaf curling from some other reason. It could be that there are cabbage aphids feeding on those leaves. That can cause leaf curling. C is due to boron deficiency. And then D is clubroot. Some folks wanted or indicated answer C. And this might be a little tricky because the browning could look like scarring, which is present on cauliflower heads when Swede Midge larvae have fed on it, but it wouldn't be internal as this picture shows. I'm going to move on to the next question, which is about Swede Midge overwintering. I'm going to go ahead and launch that. So how and where does Swede Midge overwinter? Does it overwinter as adults in trees along field edges, as eggs in crop debris, as pupae in the soil, or none of the above? Okay, so looks like nearly everyone got this answer correct, so good job. The pupae are overwintering in the soil. We talked about this quite a bit in my presentation and Christy's, and how to use that information when you're thinking about your crop rotation. So knowing where the Swede midge is overwintering is really helpful for management on a farm. Okay, a couple more questions here. The next one is on effective management. So which of the following is an effective management strategy for Swede midge? Okay, ending the poll there. And most folks were correct, and that the answer is all of the above. All of those individual practices are effective. And then our last question, we're going to revisit your willingness to try mating disruption. So we're asking that because we've been researching this for a number of years and are really curious what folks think about this. And we're doing well on time. So if anybody has any lingering questions about mating disruption, is there something that is influencing your answer here? Feel free to unmute or put that in the chat. Okay, I'm gonna share the results here. So more than half of the people said yes. So I'm curious for the folks who said no, what would be your hesitation to using mating disruption? And I invite Yolanda and Vince, if you have any other questions for the audience concerning adoption of mating disruption, you're welcome to speak again if you'd like.
Yeah, it would be great just for some audience participation if you could put in the chat kind of your reasons, either yes, why, or no, why as well. And it'd just be great to kind of enliven the conversation a little bit more. Unfortunately, we're not all in the same room, so I can't have these conversations on the side.
Yeah, so yeah, Yolanda and Vince, you are highlighting some current obstacles to mating disruption becoming commercially available, which would be kind of fine-tuning the dispenser setups, where and how many, and hoping to bring the cost down. So we're hoping that our future work will help address those things and eventually get this technology into commercial use. And your feedback on this and what might be obstacles is really helpful. So yeah, before folks sign off, we do have the sign out survey on Qualtrics for pesticide credits. So again, in order to receive the New York State pesticide recertification credits, we're expecting folks to have signed in at the beginning of the presentation and then sign out on the link that's shown here as a QR code in the chat. And last chance to send me your pesticide license photo. You can text or email it to me. Okay, good comment in the chat on mating disruption, Yolanda, if you want to address that. Those are some good points.
Yeah, I was just gonna type it out, but yes, I guess the idea is in the future it would be managed. I guess depending on the size of the farm and to what extent people outsource their pest management kind of activities, would be managed either by the farm manager, owner operator, or for larger farms, it would be managed by kind of a pest management specialist. So we're hoping, because it's, I guess, unconventional and kind of new technology, that we can supplement, a lot of extension education to make it seem more feasible. So basically, like, one of the possibilities is, like, the paste. We're hoping for that to be kind of OMRI listed materials, and it's in a caulk gun. So, like something that could just be, if you're a smaller farm, just a walkthrough kind of thing. For larger farms, we're looking at other ways of delivering materials as well. So currently, right now, everything we're doing with the pheromone work is just on the license of its testing kind of thing. So for all materials to be approved for use, they have to go through kind of a registration process, even as a biopesticide. We need to figure out whether it's worthwhile to even go there, whether it shows efficacy in the field before we go through that process. And with all materials, it's like you have to look at what the safety is, toxicity to bees, aquatic, individuals, for human health. All the same things that happen, for pesticide registration also apply to pheromones, because they're considered sort of biopesticides. And I will put my name in email in the chat if anybody wants to contact me.
Okay, thanks, Yolanda. We do technically have 5 minutes left on the webinar, so we do have time for more discussion if there's something else folks want to chat about. Was there any information that wasn't covered in the webinar that people wanted to hear about?
I have a question if no one else does. Touching on my earlier question, Yolanda, so I know a lot of urban growers would be interested in pheromone mating disruption. Could you touch on, so these are small farms, very mixed plantings. Maybe they have a bed of collards, a bed of broccoli, they have like individual beds, or sometimes it's even intercropping, which we don't need to talk about that. But where would you place the dispensers? Or my earlier question was, with different vegetables beyond brassicas in this system, are there thoughts on how effective the dispensers would be overall? Like, could that get in the way of the Swede Midge sensing things? Or
No, because the insects are so highly specialized on the particular chemicals. We have research that shows that the plant that might be, next to the midge, we've tried tomatoes, Swiss chard. We even use a fake plant just to see if structurally it does anything. And it doesn't affect their mating. They are
okay.
so attuned to the kind of sex pheromone of the female that's the most important thing. So now in an urban setting, I think the biggest challenge is just sort of like, everybody's got like a little patch here or there kind of thing. So actually identify where all those patches are, and the coordination among people, I think, would be the biggest challenges, actually. And so that's why I think that there's going to be sort of scaling of which approaches are appropriate for the size and scale of, like, the plantings, and what else is in the mix, for instance. Like, so some of your work on the urban gardens and farms, you can take advantage of, like, the lack of plantings, the lack of weeds, brassica weeds, to kind of use that to your advantage, because they're porous dispersers.
Makes sense.
Looks like there's one other question about barriers in an urban setting, so I think one of you could answer that.
Yeah, there are a couple of folks who asked questions about exclusion barriers, which I interpreted as exclusion fencing, which could be made out of screens, which is what folks in Canada tested at the University of Guelph a number of years ago. And I believe it was tested in Europe as well. In general, we haven't recommended that strategy because uh, they have in Guelph they set up traps at various heights, and they did find that the Swede midge could either fly over top or be blown in the wind over those barriers. So it can delay infestation and maybe reduce it sometimes. But the data doesn't show that it's a reliable enough strategy to manage just because they can make it over top of the fences.
In the monitoring project that we did, we had a site that had a really bushy fence line, like 12, 14 feet tall. And we noticed that the brassica planting right beside that hedgerow didn't have Swede midge. They kind of landed farther into the field. Um, but eventually the Swede midge in the rest of the field found, like when they started moving, they did find those brassicas. But if you had radishes or something, that's really quick. Um, and if you put a, I don't know, a 12, 14-foot fence up or something, or some kind of a solid wall, they could skip that first 12 feet if you wanted to get an early planting in, theoretically.
Yeah, thank you, Christy. We do go into a little bit more detail on summarizing the exclusion fencing and other work in the review paper. So that is a resource that folks could turn to if you want to read up more on some of those individual previous studies. And it is 11:30, the end of our webinar, so I'm going to end it here and stop the recording. And I wanted to thank all the presenters for sharing your work and for everyone who attended for joining us today and listening about Swede midge. And we popped our emails into the chat. We can include those in the follow-up email. So if you have any lingering questions or would like to collaborate on Swede midge outreach or research, feel free to contact us. We'd love to hear from you. So yeah, with that, we'll end the webinar, and thanks again. Have a good day.
Thank you.
Thank you, Elizabeth.